Right, I think we're gonna start the webinar right now. It's really exciting to have Jason today with us, who will be talking to us about virus-like intercellular communication in the nervous system. So this is like a probably 40th or 50th webinar, but I can't really remember exactly which one. But the whole purpose of this webinar series is to bring kind of fantastic researchers from around the world to give a talk about their research, and through that also get the opportunity to inspire the early career researchers as well as the next generation of scientists. So before going into the whole thing, let me give a brief bio about Jason. Jason Shepard is an associate professor in the Department of Neurobiology and holds the Jon M. Huntsman Presidential Chair at the University of Otago. He obtained his BSc at the University of Otago, his Ph.D. at the Johns Hopkins School of Medicine, and postdoctoral training at the Massachusetts Institute of Technology. The Shepard Lab focuses on elucidating how the brain stores information from the molecular level through in vivo neuronal networks and how these processes go awry in neurological disorders. He is the recipient of the Peter and Patricia Gruber International Research Award in Neuroscience, the International Society for Neurochemistry Young Investigator Award, the Chan Zuckerberg Initiative Ben Barres Early Career Accelerator Award, the NIH Director's Transformative Research Award, and is a U.S. National Academy of Sciences Kavli Fellow. Dr. Shepard grew up in South Africa and then New Zealand prior to moving to the US for graduate school. It's really, really exciting to have you, Jason, with us. And for all the audience, either on Facebook or on Zoom, I'll encourage you to send your questions throughout the webinar. And at the end, Jason will be able to address them. And especially for those of you on Facebook, I'll try as much as possible to go back to Facebook to see if you've got any questions that can be read through. Jason, it's exciting to have you, and the stage is yours.
Thank you very much. I'm pleased to be here, and yeah, I love this idea of trying to get international scientists together to have a worldwide audience. So thank you. So I'm going to talk about some of our new work that's really, I think, has a crosstalk with various different domains within science. And so hopefully there'll be something for everyone in the talk. So of course, the actual work that's being done in my lab is not done by me, but by these talented scientists. And I'll mention the specific people as I go along. So really the main question that my lab is interested in is, how do the cells in the brain store information for up to a lifetime? And when you consider the sort of cell biology and biochemical mechanisms that have to occur, there is this paradox in that most proteins, or almost all the proteins in our brains, don't last as long as a memory. They usually get turned over within hours to days. So what is that long-term substrate of long-term information storage, and how does it go wrong in various intellectual disability disorders and neurodegeneration? And we take a number of approaches. So we are looking at the intact circuit in the brain. So this is looking at how circuits in the brain and the mouse brain adapt to experience and change with learning. But it's really the connections between these cells, the synapses, that we think a lot of the changes are happening. And so we're trying to understand synaptic function and synaptic plasticity. And then, of course, we want to know what are the most important proteins and genes that are important for changing these synapses in response to learning. And so, of course, synapses are the computational units of the brain. This is just a very basic schematic showing that you have, of course, electrical activity through action potentials that induce neurotransmitter release at the presynaptic terminal. And then those neurotransmitters bind the receptors on the postsynaptic side And this leads to depolarization of the postsynaptic side and then propagation of the action potential. So this chemical transmission, we think, is the prime site of a lot of modifications that happen when you learn. And so, and of course, there's a temporal scale to understanding these processes. Those initial processes that I mentioned there, the electrical activity, of course, happens very quickly on the millisecond range. But we know that minutes to hours later after a learning experience, there's new gene expression and protein synthesis. And it's really this latest stage that we're quite interested, because it's been known for many years that if you block protein synthesis, you block the ability to turn short-term information, short-term memories into long-term memories. So that's the sort of temporal scale that we're dealing with. But of course, the other complexity here is that neurons can have up to a couple of thousand synapses. And if each one of these synapses can act on its own, there has to be a complex set of processes that replenish proteins there, traffic proteins to synapses. Another aspect of what neurons do, which is fairly unique, is that after transcription in the cell body, messenger RNA then actually gets trafficked out into these processes and can then be locally translated at these sites of activity in the synapse. And so that's another quick way of being able to change protein composition at these synapses. So just to summarize then, so we think that memory processes require new gene expression that's activity dependent. And then some of those RNAs are not initially translated, but they get out into the dendrites where they get locally translated. And then some of the functions that those proteins are important for include, structurally changing the shape of the synapse, the structural changes, and then changing the function, the strength of those synapses by changing the receptor composition and other proteins that regulate those receptors. So, of course, this all sort of begs the question: what are the key genes to study here? And I've taken a kind of a simplistic approach to this, and I've concentrated on this one particular gene called ARC. So ARC stands for activity regulated cytoskeletal protein. It's also called ARG 3.1. It was initially named and cloned by two different labs. So Paul Woolley's group at Johns Hopkins called it ARC. Dietmar Kuhl's group called it ARG 3.1. And both of those labs were looking for which genes would be turned on after learning. So this really represents that idea. So this is an in situ hybridization for ARC mRNA from a rat brain. And so this was a rat that was housed in its home cage. And this is a rat that was allowed to just explore a new environment where we know learning takes place. And so you can see just within an hour, there's really a massive induction of ARC mRNA in the cortex of the rat. And then the C-shaped structure, this is the hippocampus of the rat. And if you look at the blow up here, you can actually see that this is the cell body layer, but there's also a lot of RNA in these parts of the hippocampus, and those are those dendrites that stick out. So this was one of the first examples of this trafficking of RNA from cell body to the dendrites. And this observation sort of led to the idea that maybe this gene was important for learning and memory. And so Paul Woolley and Ozzie Stewart then went on to show that not only can you induce expression of ARC with learning and experience, but you can actually stimulate the hippocampus. And if you stimulate it only in one particular synaptic layer, you can actually see trafficking of the RNA to the active synapses. So really saying that there's a lot of fine tuning of where you want this mRNA to be made and to be trafficked. And that also obviously suggested that there would be local translation occurring. And so I like this experiment from Kim Huber's lab, where they took those hippocampal slices that you can record from, and then actually severed the dendrites away from the cell body, added a drug that's known to induce translation. This is a metabotropic glutamate agonist, the group 1 agonist, and then waited a few minutes and then stained for ARC protein. And so you can see immediately that there's an increase in ARC protein, even in these dendrites that were severed from the cell body. So really definitively saying you can get rapid translation in these dendrites in response to activity. So we know that the signaling pathways that control protein synthesis in synapses are extremely important for just normal cognition. So all the red boxes here are human mutations or known human mutations that cause intellectual disability or autism spectrum disorders. And so the details here are not important, but what's key is that they feed into, we think, only a few proteins that are really important for doing the work in response to these signaling pathways. And so the idea is that if we can understand what this protein is actually doing, it will give us insight into these really complex processes. So just a little bit more information about ARC. So we can make knockout mice. These are full knockouts. These mice are viable. They pretty much look normal. But in every case where we've tried to look at, and we and others have looked at memory, there's a real severe defect in consolidation and long-term memory. But short-term memory is intact, and the initial learning is intact. So if you condition an animal, for example, with fear conditioning, and probe the animal 10 to 15 minutes after the learning, they show the memory. But if you come back a day later, there's really very little evidence that they can remember that conditioning. And that seems to be the case for almost all these different kinds of learning. So it really looks like the ARC is absolutely required for long-term forms of memory. So ARC is a single copy gene, but I'll get a little bit into that later. It's a classic immediate early gene. So many of you may have heard of it because it's an immediate early gene. And that just means that it gets transcribed without new protein synthesis. So it can be turned on very quickly. And because of that, and because it's so tuned to experience and behavior, a lot of labs have used it as a marker for which circuits are active during learning, and even have used use it as a marker in other species, such as birds. But unlike some of the other immediate early genes like FOS that people may know, it is not a classic transcription factor. And we know that its expression is in dendrites and at synapses. And so we think then that it's acting in a little different way. And then in terms of where it's expressed, it's mostly in the brain. It's also expressed in the testes like a lot of other brain genes. And then in a particular type of a dendritic immune cell, which is quite interesting. And then within the brain, it's only in neurons, not in glia, or at least it's not transcribed in glia. And then the distribution, as I mentioned, is mostly this postsynaptic side. So when I started my lab, I wanted to know, a little bit more about the biochemistry of this protein to sort of try and figure out what its function was. And I like this quote from Asimov that really sort of, talks about science in a way where oftentimes we think that, if our hypotheses are not correct, that's a bad thing. But oftentimes it's the most surprising results that lead to, I think, really cool discoveries. And so I'll tell you the story here. And initially this work was led by Cameron Day, who did most of the biochemistry, and Alyssa Pastusan, who did the cell biology, and Rachel, who did the EM. And then I have a couple of people still working on this, of course, And this initial paper, this initial discovery was published a few years ago. So this whole story started with Nate Yoder, actually, my first technician. And what we wanted to do was just purify ARC protein from bacteria using E. coli. We were using this GST affinity purification protocol, which is standard way of purifying protein. So initially we thought this was great. We could express ARC, the tagged ARC in E. coli. We could then cleave ARC away from the tag. So now we had a nice pure population of protein. But then we started to get some weird results. And one of the ways to clean up these preparations is to run your protein through a column, a size exclusion column that separates out proteins by size. And so this is the chromatograph, and this is where we expected ARC to come out as a monomer. And this is where it actually came out in this fraction, this early fraction. And so this was a bit of a surprise because this fraction is the void fraction. So it's so big that it's not even getting into the column really, or not getting far down. And so we're sort of puzzled, and Nate tried a bunch of different buffers to try and get a monomer to study. And so he almost gave up on this, but Nate wanted to learn how to do electron microscopy. So he's like, well, why don't I just look at the protein that we're getting anyway? And most likely this is aggregates because maybe it's just a sticky protein. So that's what he did with the help of Adam Frost. And these were the first images. These were almost literally the first images we got from that experiment. And so if protein under EM, so this is negative state EM, if protein was aggregating, it would just be a black mess. But of course, what you're seeing here are these very stereotyped structures that kind of look like donuts or soccer balls. And they're large. These are 50 to 80 nanometers in diameter. So obviously much, much larger than a single protein. And so this is the observation that led us to this sort of almost rabbit hole of viral biology, because basically what these structures look like are viral capsids. But to remind you, this is of course a neuronal gene or a protein that we're studying here. And so at the time, this was quite a surprise. So it turns out that if you look at negative stain EM images of ARC and purified HIV GAG protein, so this is the the main part of the structural component of the virus, you can almost not tell the difference. They're very similar. And in fact, as we were doing this work and made these observations, another group, Paul's group, had shown through crystallography that the C terminus of ARC actually does have homology to this GAG protein, and in particular this this CA domain of the GAG. And in fact, if you overlay the two, this is with HIV, there's almost no difference. So pretty amazing, because of course we're comparing now a retrovirus to a neuronal protein, and somehow this neuronal protein has really strong structural homology to a retrovirus, and we now know can form these capsids. And so of course in science we can often be siloed, and there were hints about this homology even way before we were doing these experiments. This was a paper published in 2006 in Trends in Genetics. And this was a group that was using computational approaches to predict which human genes would have homology to retroviruses. And in particular, if you look at this GAG domain, they predicted that ARC had homology just based on these computational approaches to studying these genes. So I bring this up because, of course, we're now trying to figure out what this what the relevance of this retroviral homology is, but just to say that there are other gene families here that are also predicted to have these kinds of domains. And so the biology that we discover here may not be unique to ARC. So where did this homology come from? And so we collaborated with Cedric Bachot and his student John McCormick when they were still at Utah. And they're experts in these retrotransposons, which are very similar to the retroviruses. And so the gene we're studying in mice and rats, of course, is this gene here, and it's very conserved between birds, humans, and mice. But there's no ARC gene in fish, and in fact, the closest sequence in fish, this is a coelacanth, is this active retrotransposon. And we know it's active because it still has these LTR sequences, and that's what allows these transposons to jump in and out of genomes. But it does contain this GAG domain, and we think then that the best explanation for where this gene came from is that it was repurposed from an ancient retrotransposon that was integrated into the germline of an ancestor somewhere in between fish and amphibians. So all land animals have this ARC gene. So that seemed kind of straightforward, but what was, I guess, puzzling still at the time was that we also knew that there was a fly homologue of this gene of ARC. And in fact, the homology really was just in this GAG domain. So where did this gene come from? Because, you know, if fish don't have it, how does this invertebrate have it? And it turns out that this gene is another repurposed gene from a similar retrotransposon. But what's interesting here is that it's a much more recent event. So most other insect lineages don't have a real ARC gene, but they do have these active retrotransposons. So we think that this is a repurposed gene that is in the fly lineage, but it's much younger. So, you know, we're estimating 100 million years ago, this is where this gene came from, whereas the mammalian gene is 400 million years ago. But it's amazing to think that you've got this convergent repurposing of the same retrotransposon family, and what has been retained is this GAG domain. And in fact, the biology of the fly protein seems quite similar. And so I'll mention that or talk about that as well. And so this seems to be a sort of emerging theme, that a lot of these transposable elements and viral sequences in genomes can be used to generate new genes. And in fact, you know, the human genome, of course, has almost 50% of it is viral origin or transposable element origin. So a lot of the sequences that we're looking at here may actually be useful. So not being a virologist, I, of course, didn't know much about GAG. And so this is just a quick 101 on retroviral biology. And so an infected cell, an HIV infected cell, produces, of course, lots of copies of this GAG protein. Initially, it's a monomer, but there's a complex set of processes or interactions that seed this GAG protein at membranes, and usually at the surface membrane, to form the capsid itself. And as it's assembling the capsid, it preferentially interacts with its own viral RNA, the viral genome. And eventually this capsid is then encapsulating the viral RNA, and the infectious particle gets released from cells by interacting with these host cell machinery proteins called the escorts. And so this final infectious particle has the capsid, has the RNA, the viral RNA, and also this transmembrane protein encoded by the virus called envelope that confers specificity of where infections happen. So we took a very naive approach to saying, well, if that's what GAG does, how much of that biology does ARC actually also do? And so we sort of tested the life cycle of ARC and asked these four main questions. So we know that there's self-assembly into capsids, but we wanted to know, do these capsids bind nucleic acids? Do they bind RNA? Does ARC get out of the cell? And if it does get out of the cell and it has RNA, can it then transmit RNA or genetic material from cell to cell? So that's really what we tried to look at. And of course, as I showed you, ARC does self-assemble into these capsids, and they're preserved under cryo conditions. So we actually collaborated with John Briggs in the UK at the LMB, and John's lab is expert in the structure of viral capsids, and he helped us. He's helping us in collaboration with Simon Erlandson in his lab to try and get an atomic resolution structure, a 3D structure of these capsids. And as I mentioned, that Phi protein actually also forms capsids that seem similar in shape and size to these rat capsids. And for whatever reason, these fly capsids are a little more homogeneous, and so it's been easier to do a single particle cryo-EM. And Simon has reconstructed these fly capsids to atomic resolution almost, and if you look at them here, they're just they're beautiful. And really, this definitively shows that these proteins are able to form capsids that look very, very much like a retroviral capsid, even to the point where they have the same icosahedral symmetry as HIV capsids, which means the subunits form these pentamers and hexagons. So there's a lot of rich data in the structure, and we published this earlier this year. And of course, one of the big questions that we have right now is, what is the function of these capsids? And one way to tease that out is to try and find point mutations that we could use to block assembly of these capsids. And so we think we've found a point mutant that doesn't indeed have that. And so there's this wild type size exclusion column data showing that, as I showed before, that most of the protein is not coming out as a monomer, and that's what we see with these capsids. But this mutation now shifts the exclusion column data all the way to the monomer, and we see very little evidence of any assembly of this mutant. So this is exciting. We're now able to try and tease out the function of the capsid, or the capsid of ARC versus the monomer. And we actually think then that our model that we're sort of going forward with is that the protein, the ARC protein that's at synapses, is probably in its monomeric form. One piece of evidence for this was published in this paper, showing that when ARC actually interacts with these NMDA receptors, these are glutamate receptors at synapses, there's an inhibitory aspect to the interaction that blocks the ability of ARC to form an oligomer. And so I didn't really talk about this earlier, but some of the earlier work that I contributed to showed that ARC was involved in trafficking of some of these glutamate receptors at synapses. And so we found that when we overexpress ARC, you actually get a decrease in these AMPA receptors. And so this is what I'm showing here. So if you overexpress wild type ARC in a neuron, you get a decrease in surface GLUA1. But this point mutant in the capsid, we think shouldn't affect the trafficking role of ARC if that's what the monomer does, and that in fact is the case, that the this point mutant has the same ability to decrease AMP receptors at synapses, just like the wild type. So a lot of ongoing work, and so I'm going to just sort of mention the questions that we're following up on through each of these sections. And so, of course, we want to still try and figure out what the atomic resolution structure of the rat capsids are, and we're looking at that. We'd love to visualize where these capsids are actually assembling in neurons, and how they're assembling. So, what other proteins are involved in regulating that process. So, does ARC bind nucleic acids? And we had hints of this even in these purified recombinant protein capsids. And so we predicted if there was going to be encapsulation of RNA, that most likely would be ARC mRNA. And so we used RT-qPCR to look at this, and in fact we can detect ARC protein in these capsids. And if it's inside the capsid rather than stuck on the outside, you'd expect that if you added RNAs, that there would be protection of that RNA, and you wouldn't degrade it. And that is indeed the case. So when we add exogenous RNAs to these capsids, we still readily detect ARC mRNA. What was interesting from these bacterial preparations is that we also looked at sort of highly abundant bacterial mRNA to see if they were getting encapsulated, and indeed they are. So there's questions about how specific this whole encapsulation actually happens. This is just a positive control to show that if you added RNAs to, if you added naked GFP mRNA to the same test tube, it gets degraded. So the RNA is working. RNase is working. We also, of course, want to know if ARC associates with its own mRNA in vivo. So this was an experiment where we took mouse brain lysate and then immunoprecipitated ARC with an ARC antibody to enrich for the ARC protein complex, and then did RT-qPCR. Excuse me. And so we can enrich for the protein, and then what we do find is that there's enrichment for also ARC mRNA in this complex. So this says that in vivo, ARC can associate with its own mRNA. Of course, it doesn't say whether it's in a capsid or not, but it at least says that it can happen. So lots of questions here. We want to know what is the specificity of RNA binding? What kind of RNAs can be encapsulated? How does ARC bind these RNAs? So, which part of the protein? And so this is a ongoing study. And then something that sort of that I can answer in the questions afterwards, but we're very interested to actually apply these capsids, or use these capsids to potentially do gene therapy and deliver exogenous cargo, and that's something that we're pursuing. So ARC forms capsids. It can bind RNA, but doesn't get out of cells. And of course, so viruses have taken advantage of that host cell biology, and it turns out that the way these retroviruses can get out of the cell is through these escort proteins. And these escort proteins are also very important for generating extracellular vesicles. So most cells form extracellular vesicles, and these are membrane-bound protein packets or RNA packets. And they can either get out from the cell through this sort of direct budding, or they go through these multivesicular body compartments through an endosomal compartment, and then get released by the MVB fusing with the membrane. So these extracellular vesicles have become quite interesting in various processes. And of course, we think that one aspect of these exosomes or extracellular vesicles is to signal from cell to cell. But there's also evidence that these vesicles are important for eliminating toxic proteins from cells, and then allowing other cells to degrade those proteins. And so many of these misfolded proteins that are involved in neurodegenerative diseases have been implicated in have been found in these vesicles. So we thought that since viruses get out and they have a membrane around them, that if ARC is getting released, it would also have a membrane and it would look like an extracellular vesicle. And so we looked at that fraction. And the way we did that was we would take primary cultured neurons and then take out the media and fractionate the media using some ultracentrifugation steps to get that extracellular vesicle fraction. And indeed, when we do that, we can see ARC being released into the media, and it's in this fraction, and it's activity dependent, because if we added bicuculin, which inhibits inhibitory cells and raises the activity in these primary culture neurons, of course, we see more ARC protein in the cells, and we see more ARC being released. So ARC does get out, and we think it's getting out in an extracellular vesicle. In that same fraction, we can also identify ARC mRNA that's protected from exogenous RNAs again. So it's inside a vesicle or it's inside the capsid. And then one of the challenges right now is trying to figure out what kind of vesicle it's actually in. So this is one of the ways you can do this to sort of separate out different kinds of vesicles is by size and density. And so these are mini exclusion columns. And again, with activity, we can see robust ARC being released into different fractions. And so now we're trying to figure out what other proteins are in these fractions, what kind of vesicles is ARC actually generating. And we think that just like other cells, neurons are making a lot of these vesicles, and ARC is only in a few of them. And so this is immunogold labeling of some of these vesicles that we've collected and using ARC antibody. And so in this field of view, there's only 3 gold particles. And so, we're estimating that ARC is only in about 10% of these vesicles. And then if you look at the blow up, you can kind of see this high density staining around the gold particle, suggesting that there's one capsid in each vesicle. And so this is sort of some of the first evidence that we have for the capsid being inside the vesicle. So again, many questions that we're still trying to address. So we want to know, what's the pathway of release? Is it escort dependent, or does it go through these multivesicular body compartments? Of course, the $1 million question is, what is the cargo? So what is ARC actually carrying to other cells? Where is this release happening? What controls the release? And ultimately hoping to try and get higher resolution structures or images of capsids inside these vesicles. So finally, given that we've shown that ARC can form these capsids and then they bind in RNA, they get out of cells. And so the question is, can they transfer or transmit signals from cell to cell? So here we used, again, those primary cultured neurons. And what we did was we used wild type neurons for our donor cells, and we used ARC knockout cells for our recipient cells. So of course, knockout neurons don't have protein or RNA. And in these cultures, because there's a fair amount of activity, you can see even the wild type cultures a lot of ARC mRNA in the dendrites. So if we incubate these knockout neurons with the capsids, the recombinant capsids that we made, we actually surprisingly see uptake of those capsids. So this is the protein uptake. And by 4 hours of incubation, we see robust transfer of the RNA out from the capsids into the dendrites. And so this delay is interesting because we think that the protein is getting in much earlier, but we think the capsid has to disassemble in order to release the RNA. And so this takes some time. So this seems to be a regulated process. If you block endocytosis, you block uptake of the protein, and you almost completely eliminate the transfer of the RNA. So these capsids are being taken up in some sort of regulated endocytosis way. But of course, we want to know, does the endogenous ARC protein act the same way? And so here we actually took those extracellular vesicle fractions and incubated those fractions with these neurons. And so again, in the knockout neurons, when we incubate with knockout extracellular vesicles, you never see any changes in protein or RNA. But when you incubate those knockout neurons with the wild type extracellular vesicles, now again, you see robust uptake of the protein. We can see ARC protein in these cells. And then again, by 4 hours, we can see robust RNA transfer. And so the kinetics here seem quite similar to those recombinant capsids. So showing that these vesicles that contain ARC can also be transferring RNA. And so finally, if the RNA is getting out into the cell and into the cytoplasm, you would predict it could get translated. And to show that, we use that trick I mentioned earlier of using this agonist that should increase translation. And when we do that, we can see further increase in protein in these dendrites that's blocked by cyclohexamide, a protein synthesis inhibitor. And so so we think then that this RNA can be accessible to be translated. All right, so just to summarize what I showed you, so my lab's a little nerdy, and so we named these ACK bars after a Star Wars character. And so these capsids that get released into these vesicles, we call ACK bars. And so we think that our working model is that they're made in dendrites, that some of the RNAs that get into the capsid are because they're also transported out into these dendrites. And then we think that that these capsids are being released from the surface membrane. And then, of course, they can get taken up by other neurons and release their cargo. I'm showing this as sort of a very neighbor to neighbor, but we don't actually know that. It could be that these ACK bars spread much further than just neighboring cells. And so, just to speculate on the function, so it's really going to depend on what the cargo is. And so, as I mentioned, ARC protein itself at synapses can regulate the strength of these synapses to these AMP receptors. And so one could consider that there's these changes in synapses that are in the neighboring cells. Alternatively, or we know potentially that there could be these small RNAs, these non-coding RNAs like microRNAs or tRNAs that could actually affect expression of proteins in recipient cells. So we're still hot on trying to figure out exactly what is happening here. And then just to remind you, we think that the fly protein, ARC protein, does the same, has some very similar biology. Vivian Budnick's group had shown that it can be made in neuromuscular junction, that another kind of synapse in flies actually gets released from fly neuromuscular junction, gets into the muscle, and then delivers some sort of cargo. And if you block this process, you block the maturation of these synapses. So really cool conservation of this retroviral biology. So yeah, many questions still to address. And of course, in terms of memory consolidation, we would love to know if this capsid biology is definitively involved in that. So again, just to thank my lab and our amazing collaborators who have really helped us in a lot of these experiments that were outside of my domain or my expertise. And then of course, our funding sources. And with that, I'll take any questions.
Fantastic. Thank you very much, Jason. This is so amazing, and it's completely taking me by surprise. Lots of questions. Yeah, I don't even know where to start from. Before we get some questions from the audience, I was just wondering, I mean, since ARC is produced in individual neurons, do you have any speculation? Why does it need to be transported to another neuron? Why the whole buzz of transportation from here to there?
Yeah, exactly. I mean, it's still a surprise to me, in fact, because most models of how memory work, talk about the changes in synapses, but they're all specific to the cell that where ARC is being made. So there's two possibilities. One is that in a circuit, we know that some cells get incorporated into the memory, but other cells do not. And we don't know what the signal is to tell those other cells don't get incorporated into this particular memory. So there has to be some sort of inhibitory signal. And so we think ARC could be doing that. So it could be, if it's affecting synapses and weakening synapses in those neighboring cells, that could basically take them out of the memory circuit. So that's one possibility. The other possibility is that this signaling pathway is not involved in memory. And in fact, it's acting as some sort of long range signal. And by long range, I mean getting out of the brain, trafficking to other organs, or it's being released from neurons and going to other cell types, other kinds of cells in the brain like glia. And so we don't know yet exactly whether that's the case, but we're testing that right now.
I mean, this is exactly the question that someone asked, that is there any specificity in terms of which cell types, neurons only that can take up ARC capsids, and how could that be mediated?
Yeah, exactly. And so we have hints that there is specificity. So if we take the capsids and put them on like Hicks cells, a kidney cell, they do not get taken up very well. We do know that they can get taken up by neurons, but there's some preliminary evidence that that they are taken up by astrocytes. We're still looking at microglia as well. So it's definitely possible that they get taken up. But in terms of what confers that specificity as well, we don't know yet. We predict that there's either transmembrane proteins on the vesicles themselves that confer specificity, or there could be an actual receptor for the capsid itself. And so we would love to know, what those are.
Yeah, lots of questions. So there's this question, it's asking, very interesting talk, if ARC capsids are also exosome dependent, what do you think is the advantage of ARC encapsulation of the cargo as opposed to exosomes alone?
Yeah, so that's also a good question. We've also been trying to think about why would you want a capsid as opposed to just having a vesicle. Mm-hmm. And of course, in the case of viruses, the capsid is there to protect its viral RNA or the DNA from the immune system, but also it confers stability of the virus. So, some viruses can survive outside the host for a long time because these capsids are so structurally rigid. So, and so at this point we're sort of speculating, but there's two, I think, really interesting aspects of this. One is that exosomes and extracellular vesicles in particular, it's not clear how you enrich for specific cargo, right? So the sorting mechanisms of the proteins and RNAs inside exosomes have remained unclear. And so here we've got a very well worked out system that can confer specificity of which proteins and RNAs are being incorporated into the vesicle. And so that's what viruses do. So that's one idea. And the other idea is then that if this is a long range signal, that the capsid would then protect the nucleic acids inside it for however long you need it before it gets taken up by recipient cells.
Fantastic. Now that you talked about nucleic acid inside it, do you think the capsid only contains nucleic acids, or do you think it will contain proteins as well? And I was wondering, because talking about this, the first thing that came to my mind is how proteins get transmitted in neurodegenerative diseases like tau, etc. Are you thinking towards that direction? And do you think this could potentially be a cargo for such proteins that are transmitted?
Yeah, exactly. I didn't have time to really talk about that, but we have a project to look at that specifically. And so we do think potentially that ARC could be involved in the spread of pathology, tau and A beta, for example, in Alzheimer's disease. And so we have some evidence that these vesicles are containing these proteins. And so there's certainly also the case that the capsid can interact with proteins, not just RNA, and pull them into that vesicle. So we're definitely we're testing that as well in terms of spread of pathology.
Fantastic. This is really, really cool. We've got another question. Is that, have you or anyone been able to come up with ultrastructural evidence for ARC capsids being formed and released in intact brains?
Yeah, another good question that I wish we could answer. It's been sort of a needle in the haystack kind of question. You know, we can see structures in neurons that look like capsids, but to really definitively say that it's ARC, you know, we need to tag the protein in a way so that we can visualize it as well. And so we've been trying some correlated light EM techniques, and one of the challenges has been, you know, to tag the protein so it doesn't interfere with the capsid assembly. And then the other thing is that unlike a viral infection, of course, where you could potentially see hundreds or thousands of capsids in an infected cell, we don't think that a neuron is making hundreds of capsids. So it is a challenge to try and sort of figure out where these things are. And so I guess the long answer is that we don't have direct evidence yet.
Great, yeah. There's a question that said, I did not get which point mutation affects the oligomerization of ARC. Could you please repeat it?
Yeah, so it's a mutation in the GAG domain in the CA domain. It's in that C-terminal part of ARC that we predicted would be important for assembly reactions.
Great. So this might be a naive question. I know we are running out of time, but so in any of these neurodegenerative diseases, has ARC been shown to become altered in any of these diseases? Do you know?
Yeah, so in Alzheimer's, there's actually quite a lot of evidence showing that you sort of get these abnormal bouts of expression, possibly due to sort of subthreshold seizures. And there's quite a lot of evidence that in Alzheimer's there's hyperactivity initially and in circuits and sort of almost like epileptic seizures. And in mouse models, people have shown that results in really high levels of ARC and other activity-dependent genes, and they get turned on and they're on for a long time, and then then it becomes actually hard to induce them normally with learning. So we think then that there could be this sort of cycle where if ARC is involved in eliminating toxic proteins, and you get high activity levels at some point, you could overload the system and you get too much release of ARC, and then that's how you get spread. And in fact, I was involved in a study that was published in Cell in 2010, I think, that showed that if you cross the ARC knockout neuron in mice to an Alzheimer's line, an APP presenilin line, you actually get a reduction in plaque pathology. So there's hints that this is actually an important pathway in this disease, and so we're certainly excited to follow up on that.
Yeah, lots of questions to explore. Really excited for you. And I hope you get lots of money to be able to pursue those things. So in the remaining minutes, unless we get any question, I would appreciate if you could share some, your career trajectory or advice or tips or something. Oftentimes we get a lot of ECRs joining these talks. It would be nice to see and to hear how you ended up being where you are.
Yeah, no, sure. So, as you mentioned, I grew up in South Africa, and then my family moved to New Zealand when I was 14. And so I went to high school in New Zealand, and then I went to undergrad in New Zealand. And in undergrad, I became very interested in neuroscience and actually took a neuroscience major. And so I even at that point, I definitely knew that I wanted to research. and I was trying to figure out where I would do my PhD. And so one of the pieces of advice I would give people is that, try and seek out as many opportunities as you can and create opportunities. So what I did was, we actually had an exchange program with the University of California system, where my university would pay, I'd just pay tuition from my university. I'd go to one of the UC campuses, and then I could get credit for whatever I did there for my degree back home. And so that's what I did. I went to UC Irvine as an exchange student, but essentially did their first year PhD program and in the neuroscience program. And that actually allowed me to then also apply for grad school in the States. So not only did I get credit for that work and basically did my honors thesis there, but then I was physically in America to interview. And so that was sort of a stepping stone to get into the American system. So I applied to PhDs in America. But I actually also I'd also applied for a scholarship to go to Cambridge that also had worked out. So that created, just that sort of opportunity of looking for how to get to do the research that I wanted to do, created some opportunities. And so I ended up then doing my PhD at Hopkins, and that, I think, of course, led me to be able to stay in the States, did my postdoc, and then started my lab in Utah. So, I guess my biggest advice is that you can't wait for things to happen to you. You have to make it happen. Of course, I've had some help along the way. I've had great mentors, and I've had some privilege, but and I know that it's hard to find these opportunities in certain places, and that's what we're hoping to try and address, in that we're trying to open up science to more people in the world, and so seek out opportunities, and I guess the other main advice is persistence. You're certainly going to hit roadblocks. You're going to have failures. Things are not going to work out. And so you just have to, get over those hurdles and be persistent in your career.
Fantastic. Thank you very much, Jason. It's such a great talk, and the advice you gave also very fantastic. I'm sure people who listen or who will watch the video later on will also find it very useful. Thank you very much for honoring the invitation to give a talk to us today, and hopefully you tell us more about this research in the next year or so, when you've got some more exciting findings.
Thank you very much.
To all the audience and our other collaborators who help us organize this webinar series, thank you very much, and I see you in the next webinar.
Thank you.